as bioassay for target gene effect. What in by bioassay? Bioassay is an analytical method to determine concentration or potency of substrate by its effect on living cells or tissues. Target gene is target DNA, foreign DNA, passenger DNA, exogenous DNA, gene of interest or insert DNA that is to be either cloned or specifically mutated. Gene targeting experiments have been targeting the nuclei and this leads to gene knockout. A gene knockout is a genetic technique in which one of an organism's genes is made inoperative. For this purpose, two types of targeting vectors are used. They are insertion vector and replacement or transplacement vectors. Insertion vectors are entirely inserted into targeted locus as the vectors are linearized with a homology region. Here, homology region refers to the situation where nucleic acid or protein sequences are similar because they have a common evolutionary origin. Initially, these vectors are circular but during insertion become linear. It leads to duplication of sequences adjacent to selectable markers. The replacement vector has the homology region and it is collinear with the target. This vector is linearized prior to transfection outside the homology region and then sequentially a crossing over occurs to replace the endogenous DNA with the incoming DNA. What in by transfection? Introduction of foreign nucleic acids into cells by non-viral method. If it takes place by viral method, that is called transduction, isn't it? That you have studied in 11th standard. Next one is genome sequencing and Plantgenomeprojects. What in by genome? The whole complement of gene that determine all characteristics of an organism is called genome. The genome may be nuclear genome, mitochondrial genome or plastid genome. A Genome project aims to discover all genes and their function in a particular species. Such genome projects have so far been undertaken in Chlamydomonas, a kind of algae, Arabidopsis thaliana, it is commonly called tail crush, then rice and maize plants. Genome content of an organism is expressed in terms of number of base pairs or in terms of the content of DNA is expressed in C value. C value is the amount of nuclear DNA in the unreplicated gametic nucleus irrespective of the ploidy level of the species. Have you heard about the term barcode? You might have seen in all books barcoding and also in items you buy in supermarket. This will reveal the identity of the book or item as well as details like price. Similarly, barcode in genetic term refer to the identity of the taxon based on its genetic makeup. In practice, it is an optical machine readable representation of data which describes about the characters of any plants or any objects. Next, let's discuss about evolutionary pattern assessed using DNA. In recent years, the evolutionary relationship between different plant taxa is assessed using DNA content as well as the similarities and differences in the DNA sequence. Based on such analysis, the taxa and their relationship are indicated in cladogram. What in by cladogram? Already you have studied in 11th standard. A branching diagram showing the characteristic relationship between a number of species. Such cladogram will show the genetic distance between two taxa. It is also showed antiquity or modernity of any taxon with respect to one another. Next one is genome editing and CRISPR-Cas9. Genome editing or gene editing is a group of technologies that has the ability to change an organism's DNA. These technologies allow genetic material to be added, removed or altered at particular locations in the genome. Several approaches to genome editing have been developed. The recent one is CRISPR-Cas9, which is short form of clustered regularly interspaced short palindromic repeats and CRISPR associated protein 9. The CRISPR-Cas9 system has generated a lot of excitement in the scientific community because it is faster, cheaper, more accurate and more efficient than other exciting genome editing methods. Rice was among the first plants to be used to demonstrate the feasibility of CRISPR-mediated targeted mutagenesis and gene replacement. The gene editing tool CRISPR can be used to make hybrid rice plants that can clone their seed. Imtiaz Khan and Venkatesan Sundaresan and their colleagues reported in a new study which clearly shows one can re-engineer rice to switch it from a sexual to an asexual mode. Next one is RNA interference. Before learning RNA interference, we should know the central dogma of a cell. The central dogma of the cell is DNA transcribe mRNA. DNA here replicates itself, then it undergoes the process transcription and form mRNA. This mRNA translate protein. mRNA is the main RNA involved in protein synthesis. Now we shall see how this RNA interference works. RNA interference is the mechanism of silencing gene expression after the gene has been transcribed. Here, RNA interference breaks the targeted mRNA. As a result, it stops protein synthesis. So, what is RNA interference? RNA interference is a biological process in which RNA molecules inhibit gene expression or translation. This is done by neutralizing targeted mRNA molecules. Simplified model for the RNA interference pathway is based on two steps, each involving ribonuclease enzyme. Usually RNA is single-stranded. There is a chance to produce double-stranded RNA in a cell. This double-stranded RNA has a capacity to produce three kinds of RNA. They are siRNA, shRNA and miRNA. siRNA means small interfering RNA. shRNA means small hairpin RNA and microRNA, that is miRNA. When these three kinds of RNA binds with mRNA, this mRNA breaks into small pieces. This process is called silencing. This process facilitated by risk factor, which means RNA induced silencing complex. The siRNA is unwound during risk assembly and the single-stranded RNA hybridizes with mRNA target. This RNA interference is seen in plant feeding nematodes. This nematode infects roots of tobacco plant and reduces its yield. If RNA interference is present in this nematode, it fails to synthesize protein, so it leads to death of this nematode. Thus the way RNA interference helpful to plants.